The queen cage is now suspended between two frames of brood in the lower hive body. Depending on the strain of bees and the nectar flows, the quantity of brood will vary from one stock to another. At this time of the year, most colonies have no brood at all. All work is done, for the time being, when the last bees have been brushed off the last honeycombs. All other stocks are now dissolved, and the populations of bees are also turned into shook swarms. To begin with, we must cage the queens. Later, the cages will be transferred to the swarm boxes. After spraying the warm funnel with water, the bees are brushed off the combs containing honey. In the case of totally empty frames, the bees are shaken rather than brushed into the funnel. Bees which had found refuge in the space of the deep floor are also dumped into the funnel to join the others in the swarm box. In the end, the box is bounced hard on the ground and the funnel is exchanged for a crown board. Before the swarm boxes are removed from the site, any bees adhering to the outside are brushed off so that they can fly to join the colony which will remain at the site. That colony will stay here for a further 3 weeks if its reserves of food permit. After that, it will also be turned into a shook swarm. The shook swarms, mostly infected with varroa, are now taken to a dark room, treated overnight with a varroacide and fed with soft candy. After this period of rest, each shook swarm made of these old honey-producing colonies will be united with a young colony made in early summer. To replace the plastic foil over the top bars, a sheet of newspaper, pierced in several places, is used. Nibbling their way through the paper, the shaken bees will join the other bees slowly, and uniting two colonies is achieved peacefully. An empty brood box is now put on the top of the newspaper, and we spray water over the shook swarm to stop bees flying up. After the removal of the queen cage, the bees are shaken into the empty brood chamber and the hive is closed up. This annual or rotational rejuvenation of colonies strengthens each healthy young stock by uniting it with an old colony after this has been freed of varroa mites. The result is that all colonies are powerful and well prepared to withstand the ravages of winter. Over the following days, we removed the empty chambers and any remaining pieces of paper. Late in autumn, the inner floorboard is removed. This ensures that in winter the bees remain cool, stay tightly clustered, and are well ventilated from below. In order to keep an eye on the varroa populations, a varroa screen with a paper inlay is put into the deep floor space temporarily. Because the colonies are now free of brood, another varroa treatment can be administered at any time. This depends very much on the initial varroa infestation and the chances of reinfection from outside sources beyond our control. Two to 3 weeks afterwards, the varroa inlays are removed and the number of dead mites is evaluated. This gives us a good picture of the extent of the populations of the parasite. After this, the colonies are largely left alone for the rest of the winter. The annual rotation system in bee management produces strong, varroa-resistant and powerful stocks which are able to overwinter and utilize all available nectar sources for an optimum honey crop the following year.